carbon-containing compounds. Coordination compounds look at transition metals. Carbon is found in period 2, whereas transition metals is found in periods 4, 5, and 6. The atomic orbitals involved in carbon are the 2s and 2p atomic orbitals. For the transition metals, it's actually the d atomic orbitals, which are the orbitals involved in bonding. Now, it turns out that these d atomic orbitals often are empty. Okay, so since they're empty, that means that those 2 electrons in that lone pair from the nonmetal can bond to that empty orbital, or can overlap with that empty orbital in that transition metal. So in organic chemistry, we looked at 4 bonds of carbon. That's the octet rule. Now, in coordination compounds, there can either be 4, 5, or 6 bonds to the metal. So in other words, you can either fit the octet rule, or it can have an expanded octet. If it has an expanded octet, that's either called the 16 electron or 18 electron rule. The geometry using VS-EPR theory of organic compounds is usually tetrahedral. For coordination compounds, if you have 6 bonds to that metal, that's actually an octahedral geometry. Okay, so note that the shape of organic compounds and coordination compounds are different. Now, organic compounds are classified by functional group, whereas coordination compounds, we look at the ligands, or the non-metals bonded to the metal. Organic compounds either tend to be white or black. Coordination compounds tend to be colored. When I was in graduate school, my research project was on transition metal chemistry, and I actually talked to my advisor. She said that she decided she would work with transition metal compounds because it was more interesting to look at colored compounds, whether they're green or blue or yellow, as opposed to always looking at white compounds. And then in organic chemistry, we look at covalent bonds. Coordination chemistry, we look at coordinate-covalent bonds. Okay, so anyway, this is more on what we talked about earlier. So again, sp and d orbitals are used. You have the expanded octet. Let's take a look at this question here. Ethylene, diamine, and EDTA are used as food preservatives. And let me draw you the structure of ethylene diamine, and we can see how this works. So ethylene diamine. Okay, so ethylene is C2H4, and then dimene's 2. And then the amine group, does anyone know what atoms are involved in an amine compound? Yeah, nitrogen. So an amine is, we'll call it this one, NH2. I'll put an R group here representing an organic compound. So ethylene diamine is this compound here. So you have so there's ethylene diamine, and I'll put 2 hydrogens here in the lone pair. Okay, so there's the ethylene diamine. Now, ethylene diamine is often used as a food preservative, so how does ethylene diamine work as a food preservative? Any ideas? Yeah, so what's actually happening is that, note you have a lone pair here, so when you have a lone pair, this can actually form a coordinate covalent bond to a metal. So if you have a metal, it can form this coordinate covalent bond. And sometimes it's these metals, calcium or magnesium, which they change the shape or denature of protein in your food, and that causes the food to go bad. Okay, so what happens here is that the ethylene diamine, it binds to certain metals, and that's how it preserves food. Now with EDTA, that stands for ethylene diamine tetraacetic acid. So what we do is we replace the hydrogens here, these 4 hydrogens, with 4 acetic acid molecules. So it goes from where this metal combined to 2 here. So ethylene diamine, note that you have 2. You can have 2 coordinate covalent bonds to metals. So that's called a bidentate. This is called a bidentate ligand. Okay, now some of you are going to go to dental school, so dentate means bite. So note that you can imagine that the two sets of lone pairs of the nitrogen is biting on the metal. Now with EDTA, ethylene diamine tetraacetic acid, that's actually a hexadentate ligand. Okay, so EDTA. Okay, so this is the hexadentate ligand. So this means that there can be 6 coordinate-covalent bonds from EDTA to a metal. Okay, so EDTA, again, works just like ethylene diamine. It can bind to metals, and it's often referred to as a sequestrant, because it sequesters the higher metals. Okay, let's go to the next question. Why is lead and cadmium or other heavy metals used in paint? Now, how many of you have painted your room or painted before? And when you're painting, what's one issue that you have when you're painting besides getting paint all over yourself?
The smell.
Yeah, besides the smell.
Drips everywhere.
Getting the paint to stick to the wall. So note that when you're painting, if here's your wall, so you want to make sure that the paint sticks to the wall. So if it doesn't stick to the wall, that means that there's the chemical forces which binds the paint to the wall aren't working too well. Okay, so how you can get the paint to stick to the wall is one way is use heavy metals like lead or cadmium. Because what happens is that the lead or the cadmium metals, it can bind to the wall or the substance which, that texture on the wall. And then the lead or cadmium can also bind to the paint molecules. Okay, so note that lead or cadmium sort of serves as an intermediary which binds not only to the paint but also to the wall. Now, what's one problem with using heavy metals such as lead and cadmium?
Waste.
Yeah, so one problem, do you want to eat lead? You always talk about kids eat lead paint. You guys want to eat lead? What's wrong with eating lead?
Sounds like a time for the movie.
Okay, so you guys know the answer to that question. Okay, so it turns out that this is more information on coordination compounds. Now, the d orbitals are involved, and then the shape of the compound depends on the type of d orbital splitting as well as the color. Now, this link here shows a tutorial on what's called crystal fuel splitting. Now, this list here tells you how the d orbitals are split depending on the atom or the group of atoms bonded to the metal. Okay, because note that they have carbon dioxide, cyanide, En for ethylene diamine, ammonia, water, hydroxide, chloride, etc. Note that CO, carbon dioxide, will split those d orbitals the most, whereas iodide will split it the least. Okay, so if you go on to this next slide, here's what's happening. If you have a free atom with d orbitals, note that you have 5 d orbitals. All those d orbitals are at the same energy. And in chemistry terms, when you have orbitals are at the same energy, they're called degenerate. Okay, so these are these 5 degenerate d orbitals. Now, when you have a metal atom bonded to 6 other atoms, these 5 d orbitals are not the same energy anymore. They actually split into 3 orbitals at a lower energy, 2 orbitals at a higher energy. And the amount of split, in other words, this difference in energy depends on the atom or group of atoms bonded to the metal. So if we would say that in that previous slide, CO, carbon dioxide, gives a large difference in energy between these 2 sets of d orbitals, whereas iodide gives a smaller splitting in energy. And how is that going to change the color of that substance? So in other words, if you have a substance, let's just do a comparison here. Okay, so let's compare two compounds. Note that the difference in energy is different. That difference in energy is going to depend on the atom or groups of atoms bonded to the metal. How is that going to affect color? So remember, way back in Chem1A, we said that we can relate energy to the wavelength of light. So we said that delta E is equal to, does anyone remember that equation? Yeah, H is Planck's constant times c, the speed of light divided by the wavelength. So let's say this. Let's say that one substance is red, the other one's blue. Which one would be the red one? Would it be, we'll call this A and B. So which one's the red one, A or B? Okay, I guess maybe, let me give you some more information, because this is from a long time ago. This is from a long, long time ago at Chem1A. So if we look at the electromagnetic spectrum, and then, for example, if you have like radio waves, let's see, radio waves and infrared, and then visible, and then UV, and then X-rays. So in the visible, if we go from red, orange, yellow, green, blue, and violet. Now, which one has the longer wavelength, red or violet? Red. Yeah, so this has a longer wavelength, and then violet has a short wavelength. And in terms of energy, if it has a long wavelength, does it have a large E or a small E? Small energy. Yeah, so long wavelength means a small energy, and then a small wavelength means a large energy. So we're going back to our picture here. Which one is the red? Yeah, so the red is A or B? Who says it's A? Yeah, so red is A, and then B is blue. Okay, so really, you can actually design specific colored substances by changing the atom or group of atoms bonded to that metal. And that's what is actually done when you're looking at that cobalt chloride compound, that dry erase. Again, what you're doing is you're changing the atom or group of atoms bonded to the cobalt, and that changes the color of that substance from blue to pink. Okay, and then here's some metals found in biology and medicine. So I found this article in Chemical and Engineering News. The metals found in microbial metalloproteins are vanadium, manganese, iron, cobalt, nickel, zinc, molybdenum, tungsten, lead, and uranium. So there's quite a few different metals found in these microbial metalloproteins. So we know that iron is found in our body. Where is the iron found in our body?
Blood cells.
Yeah, blood cells. Turns out that cobalt is the metal in vitamin B12, I think. One of those vitamins. And then there's these We have some copper. I didn't put copper there, but there's these copper-containing proteins too. So if you're interested in these other elements, you can look at this link here. What are trace elements? So you can take a look at this link. But anyway, Bruce Ames, who is famous for this test for carcinogenicity, which he developed back in the 60s, his current area of research are involved micronutrients. So he believes that micronutrients prevent disease. So some micronutrients include folic acid, vitamins K and D, and selenium. Selenium is an example of one of those minerals which are important for us to ingest. He also thinks that people who are obese because they don't get enough micronutrients, so they continue to eat bad food, which don't contain enough nutrients, which will actually make them feel healthier. And then what's the role of metals in the body? You can take a look at this link. And note that these metals are often sometimes used as cancer drugs. For example, this compound cisplatin is used as a cancer drug. And let's go to this last slide here. So iron is found in heme proteins. These heme proteins include hemoglobin, cytochrome, catalase, and peroxidase. And a 70-kilogram human has about 3.7 grams of iron, which 70% is in hemoglobin, 30% is in ferritin. Now heme, and these heme proteins,